Bismillah ar-Rahman ar-Rahim. Assalamu alaikum everyone. This is our third lecture of biochemistry in the blood module. In this lecture we will be covering an array of disorders collectively known as hemoglobinopathies. So what are hemoglobinopathies? Basically they are disorders of human globulins. Hemoglobinopathies are the most common genetic disease group in the world. 5% of people are carriers, so that's really a huge percentage. Now, if you look at the map, notice how most of these hemoglobinopathies concentrate in the old world, specifically in the Middle East. So, moving on, there are different ways of classifying hemoglobinopathies depending on the outcome of the disorder itself. So the first type is what is known as qualitative abnormalities or qualitative hemoglobinopathies. These are basically, they result from mutations in the globin genes, resulting in the formation of different structural variants or mutants. And there are huge number of mutations that have been identified so far. So it has been like over 700 different types of dysfunctional or mutated, I should say, mutated globin genes, globin mutations. So the second class is what is known as quantitative abnormalities, where basically there is no mutation really in the globin, but the amount of globin chains produced is reduced, it's affected. So normally the ratio of alpha to beta globin chains should be 1:1. In these cases, the ratio is altered, it's not equal. And these are collectively known as thalassemias. Then we have a third type, third condition, known as hereditary persistence of fetal hemoglobin, whereby there is no switch between gamma to beta. Rather, what you have here is continuation of the production of the fetal globin. All right, so let's start now with the first class of abnormalities, the qualitative abnormalities, qualitative hemoglobinopathies. Again, they can also be classified into different types according to where the mutation takes place and or the effect of such mutation. So mutations can take place on the surface. Usually such mutations really do not have a huge effect on the structure of hemoglobin. An example is a type of hemoglobin known as Hbe. We'll talk about that in a second. An exception is probably the HBS, which is the sickled hemoglobin, or the one that causes the sickle cell anemia. But if mutations take place internally inside the hemoglobin molecule, then we would have a huge effect, a symptomatic effect. These mutations results in the production of unstable hemoglobin. They can also affect interaction with heme. They can affect the affinity of oxygen binding to the heme molecule. So usually these hemoglobin molecules are unstable. They get clustered and aggregate together, forming what is known as Heinz bodies. Again, we'll talk about this. Eventually, they cause having unstable hemoglobin results in hemolytic anemia, where red blood cells rupture and they do not survive for a long time. An example is a hemoglobin molecule known as Hemoglobin Hammersmith. Mutations can also take place in the globin proteins, whereby there is a formation of what is known as a methemoglobin, and methemoglobin is the globin molecule that has a heme that is bound to iron in the ferric state, that is Fe3. That results in inability of the globin molecule to carry oxygen, and that results in cyanosis. We can also have mutations that affect the interaction between the different chains, particularly the alpha 1 and beta 2 chains, and that can affect oxygen affinity. They can either increase affinity or decrease affinity. Mainly, affinity increases, and that causes a condition known as polycythemia, and it results from the inability of the hemoglobin molecule to deliver and release oxygen when it gets to tissues. So let's talk about the first one. We talked about HBS earlier in the introductory biochemistry course. So just to refresh your memories, it's caused as a result of a mutation in amino acid number 6, whereby we have a change of amino acids from glutamate to valine. Glutamate is a negatively charged amino acid. Valine is a non-polar amino acid. The hemoglobin is designated as HBS, and it contains, it's designated as alpha 2 beta S2. Now, what happens with this hemoglobin molecule is that the defective hemoglobin molecules aggregate together, forming an array, a fiber of hemoglobin molecules, especially in the deoxygenated state. That results in deformation of the red blood cell. It causes hemolytic anemia. The red blood cells cannot survive for a long time. Their half-life is shortened a lot, from 120 days to about 17 days. Now, so the cause of this clustering or aggregation of hemoglobin is a result of repeated cycles of oxygenation and deoxygenases. Now, what happens again during these cycles? You have this aggregation, and deformation of red blood cells that can clog, that can aggregate as well, and they can clog capillaries, resulting in inability to deliver oxygen to peripheral tissues, resulting in local hypoxia. Now, failure to deliver oxygen to tissues would lead to eventually organ failure, particularly in kidneys, heart, and lungs. So the question is, what is the molecular cause of this clustering? So what happens here, especially in the deoxygenated state or in the T state of hemoglobin, so what happens here is that this valine creates a protrusion, as you can see here. So normally this deoxyhemoglobin also has a hydrophobic pocket in the beta 1 chain. So right here you have a hydrophobic pocket, and in the beta 2 chain where the mutation takes place, you have this hydrophobic amino acid, non-polar amino acid, protruding outside. So what happens is that when you have the formation of deoxyhemoglobin, that results in insertion of this hydrophobic amino acid into the hydrophobic pocket. So you have this continuous formation of these clusters of the of hemoglobin in the T state. All right. So there are different variables that can increase sickling or increase the formation of these aggregates. So logically, anything that leads to the formation of deoxygenated hemoglobin would do that. So conditions like decreased oxygen pressure, like living or spending time in high altitudes, increase the pressure of CO2, decreased pH, increased the level of 2,3-bisphosphoglycerate. We talked about all of these earlier, as well as dehydration. So anything that results in facilitating the formation of hemoglobin in the T state would increase sickling. Now, it's not all bad, because we have something known as sickle cell trait. This is a condition in individuals who are heterozygous. That is, they have one good beta chain and they have one bad sickled globin. And these individuals are really normal. Their red blood cells do not have the same half-life as normal individuals. They are shorter. But this is an advantage because these individuals are protected from being infected with a parasite known as Plasmodium falciparum, which causes malaria. Now, notice in this map here you have the distribution of sickle cell allele or the sickle cell trait in Africa, and here you have the distribution of malaria to the right. You can see that there is really this overlap between the two. Now, the reason is that it's really advantageous for individuals who are heterozygous because they do not get infected with this parasite, they do not get malaria, and they can survive for a long time. Now, the question is, what is the cause? Why is it advantageous? There are different hypotheses, but one of the strongest hypotheses is that red blood cells have a shorter half-life that is shorter than the life cycle of this parasite that is needed to develop the mature parasite. So this parasite cannot develop and mature as a result of that. Okay, I hope it's clear. So let's talk about another type of hemoglobin molecule known as hemoglobin C. Now, with hemoglobin C, we have a mutation in the same exact position as the HbS. That is the 6th position. Amino acid number 6 is replaced. Instead of glutamate, now you have lysine. That is, instead of a negatively charged amino acid, we have a positively charged amino acid. Now, these molecules, HbC, hemoglobin, is less soluble. It can crystallize in red blood cells. You have red blood cells with deformed shape, as you can see here. HbC also leads to water loss, resulting in smaller red blood cell and increasing the concentration of the hemoglobin protein. In terms of severity, it's not really severe. It's mild. It can result in minor hemolytic disorder. Now, individuals can actually also have what is known as HBsC. That is, you have one chain mutated with sickled disease, sickled hemoglobin, and you have the other chain having a mutation for HbC. So that results in this condition right here, HBsC. Now, these individuals, amazingly, there is no clinical consequence. The severity depends, it's variable, it depends on the individual, but really you have mild hemolytic disorder in these individuals. Now, let's talk about HbE. Now, HbE is relevant for us because it's common in Southeast Asia. So if you have someone in the house from Indonesia, there's a good chance that they might have hemoglobin E. Now, there are no clinical symptoms whatsoever, so these individuals are normal. HbE results in from a point mutation in codon 26. It changes glutamate to lysine, but that does not really change the structure of the molecule. But what you have here is it has both quantitative and qualitative characteristics. So it is qualitative because the amino acid is mutated, and it's quantitative because the amount of hemoglobin produced in present in red blood cells is 60% of the normal amount of beta globin. So what does this mutation do exactly? Well, the mutation takes place in exon 1. So exon 1 can be spliced and ligated or connected to exon 2, and exon 2 with exon 3 producing a normal globin protein, beta globin protein. Now, what this mutation does is that it creates a cryptic splicing site. In other words, there's a chance that that this mutation would cause an alternative splicing whereby a portion of exon 1 is missing. That results in the formation of a defective globin protein, beta globin protein, that is eventually degraded. So you have reduced amount of beta globin in these cells. There's a condition known as Hb Hammersmith. Now, with the Hb Hammersmith, what you have here is it's a mutation in the globin protein that results in reduced affinity to oxygen. Now, the mutation takes place inside the globin protein. So you have a change of phenylalanine into a serine. Phenylalanine is a hydrophobic amino acid. Serine has a hydroxyl group. So the hydrophobic pocket or the hydrophobicity of the heme-binding pocket is reduced as a result of having a serine. That also changes the positioning of the heme molecule. So binding of oxygen is, the affinity of oxygen binding is reduced. That results in eventually in cyanosis. That is inability to deliver, as a result of inability of delivering oxygen to peripheral tissues. Now you can have mutations that can also affect the T and R state. That basically results from mutations that affect the interaction between the chains, particularly alpha 1 and beta 2. So right here you can have substitution, for example, of histidine 146. We talked about histidine 146, which is the very last amino acid in the beta chain. If it changes to lysine, that produces a hemoglobin that is more often in the R state. Now you can have also mutations that can affect hydrogen bonding between the chains that can either favor the formation of the R state or the T state, like Hb Kansas, for example. You have a mutation that stabilizes hemoglobin in the R state, or Hb Yakima, whereby it's a hemoglobin that is stabilized in the T state. Okay, so let's talk about methemoglobin and methemoglobinemia right now. Now, methemoglobin, as I said, is a hemoglobin molecule that has iron in the ferric state, that is Fe plus 3, rather than the ferrous state, that is Fe plus 2. Now, what happens here, so heme in this case is known as hemin. Now, in this case, hemoglobin cannot bind to oxygen because of this iron. Now, this hemoglobin is known as methemoglobin, and the whole condition is known as methemoglobinemia. Now, there are two reasons that would keep iron in the ferrous state in hemoglobin. The first factor that prevents oxidation of iron is the hydrophobic environment surrounding heme. We talked about that before. Now, the second reason is that there is an enzyme known as methemoglobin reductase. It's also known as cytochrome B5 reductase, same thing. So, this enzyme, what it does is that it utilizes NADH to reduce iron from Fe plus 3 to Fe plus 2. Okay? Now, people with methemoglobinemia, you can look at the pictures, you can see how this baby has blue color. It results from having hemoglobin in the with iron in the ferric state. And their blood, if you look at it to the right, it looks brownish, so it looks more like the color of chocolate. This is a main characteristic of a person having methemoglobinemia. So the question is, what does cause the production of methemoglobin? Well, there are different causes or different reasons. One of them is that you can have mutation in the globin protein itself. So you can have a hemoglobin molecule, in this case known as hemoglobin Boston, whereby distal histidine is mutated into a tyrosine. So remember, distal histidine is the histidine that stabilizes the interaction of oxygen to heme. When it is mutated to tyrosine, this stability of oxygen binding to heme is reduced. Well, not only that, but tyrosine also has a hydroxyl group. And this hydroxyl group would result in the oxidation of iron from the ferrous to the ferric state. Something else that is important is that tyrosine also attracts water into this hydrophobic pocket. Well, it's less hydrophobic than before. Water is attracted into the pocket, and that also facilitates the oxidation of iron. You have another hemoglobin molecule known as hemoglobin E It. In this case, the proximal histidine is replaced by a tyrosine. Now, you can have also deficiency of the reductase enzyme that results in the formation of the methemoglobin as well. Now, there are certain drugs, as well as if water contains nitrates. These are factors, external factors that can result in the formation of methemoglobin as well. So what is the treatment for methemoglobinemia? Give person methylene blue. Why? So look here, as I said before, that this enzyme requires the cytochrome B5 reductase, requires having a reduced cytochrome. This reduced cytochrome would help in the reduction of iron from the ferric to the ferrous state. Now, this oxidized cytochrome can be, or the reduced cytochrome can be regenerated from the oxidized cytochrome by NADH that is produced from the glycolytic pathway. Now, if there is a problem with this enzyme, if there's a mutation in this enzyme, and these patients are given methylene blue, blue is converted into what is known as leukomethylene blue by another enzyme, and this requires NADPH. And NADPH comes from the hexose monophosphate shunt that converts glucose to 6-phosphogluconate. Right? So leukomethylene would function similar to the reduced cytochrome, helping in the reduction of iron from the ferric to the ferrous state. All right. So now let's talk about quantitative abnormalities, or they're known as thalassemias. So thalassemias are actually the most common human single gene disorder. They are caused as a result of disturbance in the ratio of alpha and beta chains. Look at the distribution of thalassemias and globally. You can notice how it's really concentrated in the Middle East as well as Africa. So it is a problem actually in Jordan as well. Now, here's a reminder of the biosynthesis of the globin proteins. Remember that we have the alpha globin produced early on in the fetal stage, and it continues throughout life. Gamma starts alongside the alpha globin, and the beta starts early on, but it peaks after birth, 12 weeks after birth. Okay, so remember that. So any defect in the production of alpha would be symptomatic early on, and any defect in beta, in the production of the beta globin, would be apparent after birth. So alpha thalassemia, basically, as I said, it's you have underproduction of the alpha globin chains. Now, because we have reduced amount of alpha globin in cells, you have more beta chains, and that results in the formation of a homotetramer of beta. These are known as HbH. So HbH is a homotetramer of beta. Now, these do not have the ability to carry oxygen. They have reduced capacity, oxygen capacity, and they can aggregate, as you can see here in these two images. They can aggregate, forming these clusters, collectively known as Heinz bodies. Now, these Heinz bodies can bind with the plasma membrane. They can disrupt the plasma membrane of red blood cells, resulting in hemolytic anemia. So what is the main type of mutation that results in alpha globin? Well, it's mainly deletion, rarely point mutations. Now remember this really important point. We have 4 alleles of alpha in a person's genome. And that results in variable severity. Now, there are cases where you have deletion of 1 of the genes, 2, 3, or 4. So there are different probabilities, different levels of severity. If 1 is deleted, it's really no big deal. The person is known as a silent carrier. If 2 are deleted, the clinical symptoms, basically they're mild. If 3, then you have what is known as higher tendency to form HPFH, which is the homotetramer of beta globin. If 4 are deleted, then this is really severe, since alpha is produced early on in the fetal stage. Okay. So if 4 alpha genes are deleted, that results in the formation of a homotetramer of gamma chains. This is known as Hb Bart. And Hb Bart has no oxygen carrying capacity, resulting in oxygen starvation. And this condition is known as hydrops fetalis, and the baby would be born dead or it dies shortly after birth. Okay. Hemoglobin H disease is characterized by the formation of HBH. Now, which is, so basically HBH disease is when 3 of the alpha chains are deleted. That causes mild to moderate hemolytic anemia in adults. You have high level of HBH in these cells, and the disease is not fatal. Now, you have 2 other conditions that are really minor or even asymptomatic. The minor condition is known as minor alpha thalassemia. That's when you have 2 of the alpha genes inactivated or deleted. So individuals are asymptomatic. And we have silent carrier when 1 of the genes is deleted. Okay. Again, these are the different categories or different genotypic types of alpha thalassemias. You have the silent carrier thalassemia trait, you have the HBH disease, you have the alpha thalassemia major when all 4 are deleted. Okay. So let's go over to beta thalassemias now. So beta thalassemias, from the name, this is it's caused from defect in the production of the beta globin chains, and that results from point mutations in the gene itself, in the coding region itself, or mutations within the promoter region, in the translation initiation codon, splicing positions or splicing locations, that is, the protein is not, the mRNA is not spliced properly, or in the polyadenylation termination signal. So in this case, you would have a messenger RNA that is really long, and this messenger RNA cannot be processed properly. It cannot be transported outside the nucleus and so on. Now, having a high level of alpha globin chains would result in the formation of alpha globin homotetramers, and these are really insoluble. They result in a premature red blood cell destruction in the bone marrow and spleen. Now, we have different types or different classes of beta thalassemias depending on the severity of the disease. You can have something known as beta thalassemia major. In this case, 1 of the genes is not expressed whatsoever. So you have only one allele that is working. These individuals suffer from severe anemia. They require blood transfusions, and the problem with blood transfusions is that you have, these individuals would have iron accumulating in organs, and that results in tissue damage, particularly in heart, liver, and the pancreas. And usually they die early on, in their early 20s. You also have something known as beta thalassemia minor. Here you have production of good amount of the beta globin. Usually these people are asymptomatic. Okay, asymptomatic. So they are classified also into different types depending on severity. If you have beta null, there is no production whatsoever from the beta gene, beta globin gene. This is known as beta thalassemia major. These are the phenotypes: severe anemia, transfusion dependent, there's iron overload, and so on. Now, beta thalassemia minor, also known as beta thalassemia trait, here you have one good beta globin gene, and the other one either is null, that is, it's not functional whatsoever, or there is reduced production of the beta globin gene. These individuals are asymptomatic. Now, we have something also known as beta thalassemia intermedia. That is, you have something in between minor and major, where you have both genes having reduced expression, or one of them having reduced expression and the other one is null, or when you have a condition where you have HBE and the other allele is either having reduced expression or no expression whatsoever. Okay. Finally, we'll talk about this condition right here, also known as hereditary persistence of fetal hemoglobin. These individuals fail to make this genetic switch of expression from the gamma globin gene to the beta globin gene, so they continuously make the fetal hemoglobin, HbF. Now, these individuals are asymptomatic. They are normal. They do not know that they have any condition unless they do a test, a hemoglobin test that we will talk about in a few seconds. And they do not really have an advantage over other people. That is, they do not deliver oxygen more efficiently than other individuals. But the beauty of understanding this genetic switch is that if we understand the transcription factors or the genetic components responsible for making the switch, that can help us in treating beta thalassemia. So if we induce the production of the gamma globin in individuals, adult individuals with beta thalassemia, that can solve the problem of having reduced production of beta globin chains. So this is research that is ongoing. So now let's talk about testing for hemoglobin, type of hemoglobin. Specifically, we'll talk about qualitative hemoglobin molecules. And this is done using electrophoresis. Of course, molecular technologies are utilized like PCR and sequencing, and we talked about and restriction endonucleases, genetic fingerprinting. We talked about all that before, but this is the case where we can use electrophoresis. Now, remember with the electrophoresis, proteins and molecules migrate according to charge or size, but in this case we're dealing with charge. Okay, and when you have a mutation that causes change of amino acids from a charged amino acid to an uncharged amino acid, that would cause the overall change in the isoelectric point and the total net charge of proteins. Okay, so that's the idea here. Now you can read this on your own. But the idea now is that if we look at here, we have a gel where we have migration of different proteins. Now, lane number 1 is a standard. That is, it's a sample that I know it has a combination of 4 major hemoglobin molecules. We have the HbA, HbF, HbS, and HbC. Now note how HBC migrates faster towards the cathode, whereas HbA migrates very slowly and it stays near the anode. HbS is halfway as well as the HbF. Now the reason is that, remember with the HBC we have a mutation that changes a negatively charged amino acid, which is glutamate, to lysine, a positively charged amino acid. And that's why you have HBC traveling closer to the cathode. Now with the HbS we have a mutation that changes a negatively charged amino acid to a nonpolar amino acid, glutamate to valine, and that's why it travels away from the anode. And then we have the HbA that has more negative charges than the rest. So you look at lane number 2. Here we have only one band that migrates similarly to the HbA protein standard. So this is a normal individual. Now lane number 3, you have two bands right here representing HbA and HbF. So you can say that this person is a neonate. And you look at lane number 5. Lane number 5 is the standard. Lane number 4, you have one band representing that migrates at the same level as HbS. So this is a person with sickle cell disease. Now lane number 6, you have two bands, HbA and HbS, and this individual is a heterozygote for this person has sickle cell trait. 7, you have again two bands, HbS and HbC, so this person has HbSC disease. And number 8, HbA and HbS, again this person is heterozygous as well. So these are so that's the technique that can be used to diagnose a person having qualitative hemoglobinopathy. So this is the hemoglobinopathy lecture. I hope you have enjoyed it. I hope you'll do well in the exam. Good luck. Allah ma'akum.